Every morning, a group of very dedicated and skilled scientists wake up in Uppsala thinking about this. No, it's not Play-Doh. Although they have very different backgrounds, they all share a passion and a desire to one day be able to say, I was part of developing a breakthrough therapeutic. Over the next 10 minutes, I will share with you how we work at Beactica to achieve this. So our business is the generation of novel and mechanistically defined small molecule therapeutics in areas of unmet medical need. On a practical level, what this means is that we target some very challenging disease-associated proteins in totally novel ways. To do this, we've built and validated an internal drug discovery platform based on state-of-the-art surface plasmon resonance technology. In a moment, I'll share you an example of how we apply this platform. But first, I'd like to give you a little introduction to the company and our pipeline. So Beactica was founded in 2006, and from day one, we grew by establishing long-term relationships with big pharmaceutical companies and cutting-edge biotechs. They came to us because we had unique knowledge about molecular interactions and an understanding of how to use that knowledge to develop novel therapeutics. Over these early years, they provided us with some great challenges that enabled us to build and sharpen our platform. We also developed significant breakthroughs for these clients and delivered great value. So in 2014, we took a strategic decision to start building an internal pipeline of therapeutic products. And in 2016, 2 years later, we entered into our first licensing and collaboration agreement with a Korean pharmaceutical company called Dong-A. Today, our pipeline consists of 4 prioritized programs. The most advanced one is targeting LSD1, an epigenetic enzyme. I'll come back to that. We also have 2 programs that we progress in the collaboration with Dong-A. We have an immuno-oncology program targeting USP7, ubiquitin-specific protease 7. And we also have a number of exploratory targets that we are progressing and that we bring forward into our pipeline when we need to and when the timing is right. So for the remainder of my talk, I will focus on LSD1. And what you see here is a picture of a high-resolution X-ray crystallography picture structure of LSD1 that we've developed within this program. This enzyme has 2 functions that both regulate disease-associated genes. On one hand, it's an enzyme removing methyl groups from histone groups, thereby changing the expression pattern of disease-associated genes. On the other hand, it's a structural protein that helps big complexes of epigenetic proteins to stick together. Now, there's plenty of evidence showing that LSD1 should be a really good oncology target for pharmaceutical development, but in spite of more than 10 initiated clinical trials, we're still waiting for a significant breakthrough in this area. So what's the problem? Well, all the programs in clinical development are focusing on the catalytic function of the protein, which obviously have some sort of a limitation, seeing from the evidence from early clinical trials. So one thing that our platform is really good at is addressing proteins in totally novel ways. So by putting this protein to our platform, we are now developing a set of allosteric modulators targeting instead the structural function of this protein. Now, the question is that going to do the trick? So the first thing we wanted to look at was to see what do these allosteric modulators actually do in the cells. So what we see on the left is the Western blot highlighting LSD1 quantity in THP1 cells at increasing concentrations. And what we can see is a very clear decrease in the amount of LSD1 in these cells. So we're effectively mimicking the genetic knockdown that has shown all the promise in the literature. And on the right-hand side, we see a quantification of LSD1 in the nucleus and in the cytoplasm. So we can see that we are definitely mimicking this genetic knockdown. The exact mechanism, how we do this, we're still working on. It's work in progress. But we still wanted to progress in parallel and see what we could do with these compounds in cancer cells. So one of the collaborations that we've set up is with Professor Bengt Westermark's group at Uppsala University. So they were leaders in studying glioblastoma. And one of the fantastic things that they've built is a library of glioblastoma initiating cell clones. So they've taken treatment-naive glioblastoma patients and extracted tumor material. Then they've sorted out these cells into single cells and characterized all those hundreds and hundreds of single cells and identified so-called glioma-initiating stem cell clones. And we've now had the privilege of being able to use those stem cell clones to evaluate our compounds. And today I'm going to focus on two of these clones, 475 and 21271. One is a resistant clone that doesn't respond to temozolomide, the standard of care. It doesn't respond to any other drugs that they have evaluated to this on these clones so far. And the other clone is a clone that's sensitive, a stem cell clone that's sensitive to temozolomide. So if we look at what these graphs show you is the survival index at increasing concentrations of temozolomide. And as you can see, you see the expected response in the sensitive clone that dies off over time and over increasing concentrations. And the blue, whilst the blue clone is unaffected. Now, if we look at one of the compounds that did enter the clinical trials, GSK 552, you can see that that is largely not able to affect either of these two clones. Now, if we look at our compound, BA11, which is one of the compounds we're progressing here, you can see that it kills off these cells very efficiently through an apoptotic mechanism. So we needed to then make sure that we don't kill off healthy cells. So in this study that I'm showing you here, we looked at the relative confluence over time at a fixed concentration of temozolomide to the left and our compound on the right. And as you can see that on the right-hand plot in green, we have normal healthy astrocytes that are largely untouched by our compound. So these are all very exciting results, very promising. And the next thing we did was to go into animals, and we started with a single-dose pharmacokinetic study so to evaluate these compounds. And this study showed that we have good blood-brain barrier penetration, we have good metabolic stability, and we also have, although not shown in this graph, we have oral availability of these compounds. So after this, we performed a 3-week multiple dosing, increased dosing study that also showed that our compounds were very well tolerated and also that they accumulate in the brain up to concentrations that are equivalent to what is needed and what we've seen, what is needed, and that we have seen from the in vitro experiment with these cell clones. So it's all looking very promising. And right now we're in the process of establishing a unique PDX model using these stem cell clones from Bengt Westermark's group at Uppsala University. So we take them and we implant them intracranially into not-skid mice, and we're then going to evaluate the tumor load and proliferation in these models. And this is now leading up to the next milestone, which is the readout of the in vivo proof of concept study. This is going to happen before the end of the year. It's a very exciting time. We are also looking at a number of different cancer types to find follow-on indications, and the strategy moving forward to the clinic will obviously depend on what we select as the primary indication to move forward with. So to summarize, we have a first-in-class compound that showed great promise in a very difficult disease where there's no effective treatment today. And I think we're making good progress on our dream towards developing a breakthrough therapeutic for this protein. And with that, I would like to thank you very much.